Hello everyone, welcome back to my channel Xgene proteinX. In this tutorial video, I want to talk about the cell cycle regulation. So there are several proteins. These proteins are required for cell cycle transition, as well as there are some proteins that inhibit the cell cycle progression. So all of these proteins they work together and tightly regulate the cell cycle progression. So let's have a look. So there are different phases in the cell cycle: G1, S, and G2, and M phase. So basically G1, S, and G2, these phases are known as the interphase, and the M is the mitosis phase. So S is for the DNA synthesis and DNA duplicates in this phase, and then in the M phase, finally cell divide. And the G1 and G2 phase, these are just preparation phase. All of the proteins that required for S and M phase, those proteins synthesized in this G1 and G2 phase. So first is the cyclin-dependent kinase. These proteins, this is very much required in entire cell cycle process. So CDK is the family of serine threonine protein kinase, and so far there are 9 CDK identified, and out of those 5 are active during cell cycle. So this CDK 4 and 6 and 2, these are required for the G1 phase, and CDK 2, these are required in the S phase, and CDK 1, these are required in the G2 and M phase. But other remaining CDK have not been shown to have crucial role in cell cycle progression. So also there are cyclin proteins also required, which proteins associate with the cyclin-dependent kinase or CDK, and these complex are very much essential for cell cycle transition. So let's have a look at how many kind of cyclin proteins are there. So cyclin D1, cyclin D2, cyclin D3, cyclin E, and cyclin H. So these are the cyclin proteins, and out of those, cyclin D1, cyclin D2, and cyclin D3, these proteins bind with the CDK4 and CDK6, and cyclin E, it associates with the CDK2 proteins, and CDK7, it associates with cyclin H. So this CDK and cyclin D complexes are very much essential for entry in the G1 phases. So basically this CDK7 and cyclin H, this complex are essential for CDK activation. So different kind of the CDK activated by this complex. So for CDK7 and cyclin H, it phosphorylate in threonine 161 site, and then it activate CDK1, as well as it also phosphorylate threonine 172 site, and then it activate CDK4 proteins, and also it phosphorylate, this complex phosphorylate the 160 site of the threonine, and then it activate CDK2. So all of this CDK activated by this complex. So there are some proteins like WEE1 and MEAT1 kinases. So these are required for phosphorylation in tyrosine 15 and threonine 14, and then it phosphorylates CDK1, and then inactivation of the CDK1 occurs. So for those proteins, these are the inhibitory proteins, and these proteins are for inhibitory mechanism, and it inactivate the CDK1. But the CDC25C, these proteins are required to dephosphorylate this tyrosine 15 and threonine 14, and then the CDK1 activated. So there are different kind of the complex of the CDK and cyclin are required in different phases of the cell cycle. So CDK2 and cyclin E, this complex are required for G1 phase to S phase transition, and cyclin A and CDK2, this complex required for the S phase, and for CDK1 and cyclin A, this complex required for transition from G2 to M phase. So next is the cell cycle inhibitory mechanism in response to DNA damage. So basically there are two different kinds of the CDK inhibitor or cyclin-dependent kinase inhibitor. So basically this is the CDK is the main proteins. So if the CDK is inhibited, then cell cycle arrest occurs. So there are two kinds of CDK inhibitor family, INK4 family and CIP-KIP family. So in the INK4 family, there are 4 different proteins: P15, P16, P18 and P19. And in the CIP-KIP family, there are 3 different proteins: P21, P27 and P57. And this INK4 family, it inactivates G1 related CDK like CDK4 and CDK6. And this CIP-KIP family proteins, it inactivate CDK cyclin complexes. So these proteins, it inhibit the CDK4 and CDK6 alone, but this family of the proteins, they inactivate the complex of the CDK and cyclin complex. So there are basically P21, this protein, it induced by the P53 and it bind with the PCNA and then it inhibit DNA synthesis. So this occurs, so then the DNA synthesis is inhibited. So in the S phase, no DNA synthesis occurs in case of the DNA damage response. So there are the 2 different proteins like P15 and P27. These proteins expression and activation increases by TGF beta, and then cell cycle arrest occurs. Also there are normally the P53 level is lower expression, but when the DNA damage occurs, then P53 level is expressed high and then it activate P21. So upon activating the P21, it also DNA synthesis occurs inhibited. And there are 2 different proteins like BAX and MDM2. These are activated also by P53. So basically this in the P21, in the promoter region of the P21, there is one binding site of the P53. That's what P53 can regulate the transcription of the P21. So upon activating this BAX and MDM2 again it inhibit the P53 by degradation of the ubiquitination, and BAX protein, it induced apoptosis of the cells. So there are 2 different proteins like ATM and ATR. These proteins recognize the DNA damage and it induce the P53 level by phosphorylation. So when this ATM ATR induced by DNA damage response, then it phosphorylate P53 and activate the P53, and then P53 it activate the P21 by increasing the transcription of the P21, and then P21 it induces G1 to S cell cycle arrest. So also there are 2 different kinds of the proteins like retinoblastoma protein and E2F protein. These also act as a tumor suppressor protein which leads to cell cycle arrest. So also there are in case of like normally CDC25, this protein that I mentioned earlier, that this protein is required for CDK. So normally CDC25 protein, it located in the cytoplasm with complex with the protein 3 related proteins. But when the DNA damage occurs, then protein 3 group of the proteins, it separate the CDK and cyclin. So when DNA damage occurs, then this CDK and cyclin are separated or sequestered by the protein 3 related proteins. So this is the end of the cell cycle. So there are these 4 different proteins like cyclin A and cyclin B. In this protein, it has the destruction box. This is some specific sequence. And for cyclin T and cyclin E, it has based sequence. Based sequence means proline, glutamic acid, serine, and threonine. So these sequences are very much required for efficient ubiquitin mediated cyclin proteolysis at the end of the cell cycle phase. So when the cell cycle ended, then these cyclin are proteolytic degradation occurs of these cyclin proteins by this cyclin A, cyclin B, cyclin T, and cyclin A. Then cell cycle stop. So this is all about the cell cycle regulation and inhibitory mechanism. So I hope this video will be helpful. If you like this video, kindly hit the like button, share it, and subscribe my channel. Thanks.